this sort of a disease. So what we need to know, we know that the nucleus is bulky or different in size because they have excess of DNA. The number of chromosomes are not the same as a normal cell, and they have the boundaries are not defined. They can move through the bloodstream to various parts of the body, to various organs, and the muculi, the external environment they generate, has an acidic nature. So these are the things we need to know when we want to address the problem. So what is now more important, which we want to address, is to look into what happens when you have an acidic muculi. That means you have a extracellular matrix which is acidic, and what gives rise to the cancer. Let us look at the picture. Next page. Page down. Okay, just look at this picture. What we have, when you have this normal cell, there is an equilibrium maintained, then the extracellular matrix model enzyme which is present maintains the overall healthy environment. If there is a tumor cell, that is held in check. But if you have a pathological condition, what happens? You just look at this, you will see this is the epithelial cells, and here you have one cancer cell. If it is in the normal condition, it is going to be checked. But when you are having a pathological condition, what happens is the activation of the fibroblast cells occur. The fibroblasts are, if you can see, it's not moving. Yeah. These are the fibroblast cells. If you see, normal fibroblast cells are these ones. This generation of the activation of the fibroblast cell, which contributes to the abnormal buildup of the matrix, extracellular matrix. Then what happens? You have a deregulated expression of this enzyme, which is supposed to be maintaining the overall environment, and impacts the surrounding cells. What happens in the process? You see the point 2. This is the point 2, where you are having the local environment gets affected. Then what happens? The epithelial cellular transformation occurs. You see, this is the epithelial cell which is we have here, and this is where the transformation is occurring. The immune cells are utilized. Leukocytes are utilized to promote the cancer progression. So what happens? You have more, the vascular biology is getting different. It promotes tumor association, and associated angiogenesis means blood vessel starts growing in that tumor cells, and you get leaky vasculature through which the cell can migrate to different, evade the process and go to different sites, and that is known as metastasis. What happens here? The abnormal expression of the extracellular matrix model protein or enzyme creates a local niche of the metastatic state. Here you can see, using a different computer becomes difficult. Okay. So then what happens? Then angiogenic switching on is continuously left off and promotes expansion of the cancer and goes to the micrometastasis conditions. So this is how it progresses. So if you know how it progresses, then you know how to target it or how to cure it. So what are the cures which we have? We have the surgery, the chemotherapy, radiation therapy. We have the bone marrow transplant, where the stem cells of your own or somebody else's, who are related, can be used. That is the place where you generate the immune cells. So if you can change or get a transplant, then you have the therapy in that manner. You have immunotherapy. That means you improve your immune system by targeting certain immune cells, or you are, if you targeting some species which is actually evading the immune system, that species can be targeted and suppressed. That way your immunity will be better, and you can recognize the occurrence of the cancer. Sometimes you don't even know the cancer is happening, just because they are evading the immune system. Then you have the hormone therapy, like certain functionality of certain glands is going to secrete certain hormone, and if they are not functioning, some of them, some of these hormones are actually going to help in the overall metabolic activity of the cell. So if we can have this therapy, which we supply the hormone to the system, then you can have a therapy to improve the conditions or pathogen in your body. Then there is another two things, which is cryoablation and radio frequency ablation. This is cryoablation, means you have a very sharp needle which is thawed in cryotemperature, which is put in cryotemperature, and that is pricked into you, and then it is taken out and thawed, again put frozen in cryotemperature, and again thawed. This is how the needle is pricked into in the body. And radio frequency ablation is obviously you apply heat in the form of some electrical energy, and then that is how you get to kill the cells which is carcinogenic. The last one, which is supposed to be the alternate therapy which we have, is the drug delivery through nanoformulation. Now, why is this alternate therapy required? What happens is the pharmaceutical ingredient, or the drug which we are talking about, used in the chemotherapy procedure, may always not have good solubility. They may not be specific to the site where it should reach. So you use large doses of the drug, which is more than what is required, and it is obviously going to not only kill the cells which are cancerous, but it's also going to be having collateral damage in the form of the harming the healthy cells. So we want to have a better life. If we want to have a better life, we have to improve upon the active pharmaceutical ingredients we are using. And what are these? What are these which we are going to be looking into? How to control this? So we have to have a formulation where you can improve the drug solubility, make them more specific, and in the process make a process or construct a formulation which is not going to harm the healthy cells along with the carcinogenic cells or pathogenic cells. So the formulation must look through several physiological and biological barriers. It has to pass through various barriers. So delivering system. Hello. So we have to have certain criteria which is to be fixed, and those are those. Hello. Hello. Am I audible? Am I audible?
Yes, ma'am.
Okay. So the formulation must pass through various physiological and biological barriers. So they have to be armed with certain parameters. And what are these parameters they should be armed with? First, we should have the material to be biocompatible. They should have size compatibility. They should pass through the physiological and biological barriers. And the surface chemistry of the material which is carrying the drug with it, so that you have it doesn't have the specific interaction with the other organs, or introducing specific binding to the target which is required. So these are the things which we look into when we are trying to find an alternative therapy in the form of a delivery system where you deliver the drug directly to the site. So what are the things which we have talked about? We have talked about the biocompatibility, the size, It should be some drugs which we have for the cancer therapy have very poor solubility because they are hydrophobic in nature, and when you have in the body fluid, it has to be hydrophilic. So you have to make this drug so that it becomes soluble in the body fluid, and it should be a stable uh, condition so that the therapeutic concentration in the blood plasma is maintained constant. Now, what happens if you are having a carrier which is developed and you are loading the drug? If the drug loaded is not sufficient, if it is less than say some 5%, then you have loaded the drug and you are used it for the purpose which is to be targeting the cancer cells. What happens? You are actually accumulating the more amount of the carrier instead of the actual drug. So that creates a lot of problem in when you want to have this going out of your body. So this unnecessarily creates a pressure on the system where it is going to be kept in retained in the body. The carriers are retained in the body, and the load which you are carrying is very small. Now, whatever load you have put in into the delivery system, once it reaches the place where it should be effective, that time the release property should be good. If the release properties are not good, then whatever you are carrying, you cannot disperse, then you do not have the biodistribution of the pharmaceutical drug you are using. So you have to make sure that the concentration of the active pharmaceutical ingredient improves, the pharmacokinetics is improved, means that movement and the passing through the barriers is better, flowing through the system is better, and the distribution throughout the blood plasma is good. It should also ensure that it does not harm the other healthy cells, only it is going only towards the cancer cells. And it should have controlled release. What happens? There's a term called burst release, where you have sudden release of the drug and you have a sudden excess concentration, and that is detrimental. That is not going to help because you are using in excess to what is required. So it may not be uniformly distributed. So bioactivity may not be that good because the release is so fast that they are not utilized. Now, the best would be if you have the system which you have designed is actually going to the exact site. So that is known as site. If you have a specific site-specific delivery, that will be the best. And obviously, you should have the system escaping the renal system very easily. It should be cleared from the body, and it should not be captured by other microphages. Because if it is retained, then that may itself cause prolonged accumulation, may cause toxicity again. So these are the things which we look into when we are trying to design or formulate a drug delivery system. So what we are actually looking into, we are actually looking into phases where till say 1950 to initially we had this delivery system where we had oral and transdermal deliveries, and then came the second generation where you had the environmental friendly biocompatible systems, and now in the third generation we are looking into much more site specific smart delivery system. So this is what is the progress which we have. Now, when we are designing a delivery system, what are the key features we need to look into? If you want to make a smart formulation, what is the key features? What are the key features? You have to functionalize the whatever target delivery system you have taken. First, it should be biocompatible. You have to search for a material which is biocompatible. Then you functionalize the surface. Functionalize the surface with specific ligand which recognizes the the tumor specific receptors. Each of the tumor site will have specific receptors. So if you want to ensure that it is selective, then you have to have attachment on the delivery system, or which is, we say, is functionalization or conjugation with a ligand which is specifically going to go and interact with and latch on to the tumor receptor. So this ensures the site specificity, selectivity, effective cellular uptake, and minimal off-target impairment if you do not want the other healthy cells to be damaged. But if you are doing that, if you are making that unit, then when it travels, it's gone to the site where it should deliver. Now what you have to do, we have to now trigger the release. We have to have a stimuli which is going to release the drug from and separate from the carrier. So this is, we call the response system, or the stimuli response mechanism, through which the drug is going to be released from the formulation you have created. And these all is going to be some sort of stimuli which we can have depending on the cellular hematosis. It can be triggered by internal stimuli such as pH, the change in pH, the change in redox status, the change in enzymatic activity, which is going to be different for the normal cell and the cancer cells. You can also have stimuli from outside. You can have a light irradiation through which you can have the release of the drug at the site from the delivery unit you are using. You can use ultrasound, you can use electromagnetic field. So this ensures all of these response functions ensures that you have time control and site specific release of the drug. Now, if you have done that, now comes the point that whether it is reaching or not, how do you know? We should also have an optical label. So we have to design a formulation in which we have the carrier, which is conjugated or functionalized with this particular type of ligand, which is going to recognize the specific receptors in the tumor site, and it should be released on a stimuli, and it should also have a label which is going to be having a thorough output and visualization of the delivery. So, to understand this, we should understand how the targeting mechanism works. We need to understand the relation between the targeting mechanism and the physiological factors of the whatever size and surface charge of the drug delivery system, and also the pathological characteristics, path physiological, through which it is moving, of the tumor environment. Tumor microenvironment we had discussed in the beginning itself. It is important to know how the environment is. That is how we target the delivery system. So it is important to know how this tumor environment, the path through which it's covered, and the physiological factors of the drug delivery system, how do these interact and are related to the mechanism of delivering or targeting the drug. So basically, in very broad sense, not going to very nitty-gritty, the targeting mechanism can be classified in two forms: passive delivery and active delivery. And what is passive delivery? Passive delivery is accumulation of the loaded nano carriers, means drug loaded nano carriers at the tumor site due to dimensional fenestrae in the blood vessels, because you see the fenestrae means the windows or the spores in the blood vessels, which is we have already discussed that the blood vessels are much more dilated, they carry more blood, the angiogenesis is high, so more of the dilation is there, so the windows and the pores in the blood vessel is more. and they have leaky boundaries. In addition to that, they also have the lack of proper lymphatic exit. The lymphatic exit is not proper in a cancer cell. So what happens due to accumulation of this nano drug at the tumor site, this retention we call is enhanced permeation with retention effect. That means you have the accumulation of the drug carrier at the tumor site because of the porous boundary of the blood vessels, tumor blood vessels, and lack of proper uh, lymphatic exit or draining out of the lymphatic system. So this ensures the EPR effect, which is known as the enhanced permeation and retention effect. What happens? You have leaky vascularization, vascular latures, and you have leaky we have pores within the endothermal cells in the tumor vascular, and you see the angiogenesis results in more of the blood vessels, so you have leaky uh, vasculature, and then you have two junctions, which is two endothelial cells in the tumor vascular site is going to be joined. They are having a dimension of, say, around 100 to 800 compared to 1 to 10 in a normal cell. So this accumulation of the nano drug cannot penetrate the normal cell, but it penetrates the tumor cells because of the leaky vasculatures. So leaky vasculatures let uh, the nano carriers enter the tumor cells, and this is done by them appearing and in accumulation in the tumor site. So what happens is, since the lymphatic drainage in the tumor is not sufficient, you generate high pressure, and this high pressure generated in the interstices enhances the interstitial space retention of the nanocarriers in the tumor. So nanocarriers that penetrate through the fenestraries into the tumor site are internalized and retained in the tumor by what you call is the EPR effect. Then we say it is a passive delivery. And the next one, and this is how you can see the passive delivery. You see the leaky bound vasculatures of the cancer tumor sites, and this is the normal epithelial cell, and this is where you have the tumor, and this is the drug delivery carrier which you are designed, which is loaded with the drug, so they can accumulate. This is the environment, tumor environment. The drug loaded carrier enters the bloodstream, and you have it goes into the cell, and you have this uh, therapeutics or targeting mechanism starts. But only problem here is there is accumulation of the loaded drug loaded moiety in the vicinity of the tumor site. The alternative is obviously active delivery, where you have a target. The targets is the tumor site, targets the moiety targets the receptors. These receptors are overexpressed in the cell surface, and this is targeted by a ligand which is attached to the nanocarrier, and we have an interaction through the ligand receptor interaction. So we need to modify the surface of the nanocarrier in such a way that it can go and attach to the receptors which is overexpressed in the tumor sites. So the target ligand must be having the ability to bind to a specific receptor. That means it's going to be very specific, and that receptor should be overexpressed in the tumor cells or the tumor vasculature, and it should have a high surface to volume ratio. What happens if you're talking about a nano system? You're having a high surface to volume ratio. If you have high surface to volume ratio, what you are effectively doing is accessing multiple targeting moieties. So if you are able to access multiple targeted moieties, your effectiveness is obviously increased. And what are the type of targeting agents or the ligands which we are using to decorate the delivery system, so that that ligand goes and attaches to the receptor in the which is overexpressed in the cancer cell, and we can have the interaction and we can have the therapeutics generated. So what are the types of targeting agents we have? We have small molecules, antibodies. We can have antibody fragments, tripeptides like you have RGD. You can have transferrin. You can have folic acid. You can have other growth factors, nucleic acid. These are the various things, various targeting agent you can have. Obviously, the best would be to have the one which is going to be readily available, which is I have mostly used the folic acid receptor. But you have to remember, I can use the folic acid receptor as a ligand, but folic acid as a ligand for the folate receptor, but if I have to target the cell which is the tumor site, I can only target those tumor sites where the folate receptors are overexpressed. Okay, I cannot target the tumor site where the folate receptors are not overexpressed. So it becomes specific to which site you are going to go, depending on which organ the tumor is, and that tumor site is expressing which receptor is overexpressed. That will vary from site to site and tumor to tumor. So it cannot be what I can use for the breast cancer, I cannot be using that as a prostate cancer. The same folate receptor cannot be used. A same receptor is not overexpressed, so I cannot use the same type of ligand to latch it onto it. So if you see the system here, how it looks, you have the binding of the ligand, which is anchored to the nanoparticle or the nanocarrier surface, to a receptor which is overexpressed on the tumor site. And then you have the binding of the ligand anchored to the receptor, to the surface of the receptor, overexpressed in the endothelial cells. And what is the advantage? Obviously, advantage is that you have site-specific delivery, you have reduced off-target delivery of the drugs which you are using, and this obviously will increase the efficacy of the therapeutics which you are dealing with. It can overcome the process of accumulation of the nanocarriers at the tumor site, and it can also circumvent the problems of multi-drug resistance. Usually we can always think of a system to have best possible manner. We can have a system where you can have the active and the passive delivery system mechanism working together in combination. So what are the delivery systems which we have? You can see from the diagram, you have a number of them. You have the liposome, you have the polymeric nanoparticle, dendrimers, you have the carbon nanotube. So many things are there. But if I want to put them into certain class, then I can look into them and divide them into certain class. And this delivery system is not necessarily going to carry the drug. It can carry DNA, it can carry proteins, it can carry peptides. So we can have delivery of number of things to the organelles or to the tumor sites or whatever, wherever the therapeutics needs to be reached. So what are the major categories which can we can have these entire domain of delivery systems? If we can look into, we can classify them into certain categories. Like I can say, the way I have designed to speak about is organic based nanocarriers. You can have carbon based nanocarriers. You can have inorganic based nanocarrier. Inorganic structures are actually not used in as their own. They are actually always in combination with organic moiety. So usually what you are doing in a delivery system, whenever I'm talking about inorganic structure, nanostructures based delivery system, I'm actually talking about the hybrid system. Inorganic nano delivery system is hardly of much importance, because you need to coat the material, carry the material. So you need, if you have an interface of organic inorganic material, then you have the best possible carriers. So nanocarriers, what we are going to be discussing, you will see that we have the inorganic part. We design it such a way that we have the inorganic material. We functionalize that with a ligand which may be organic. We have a protective layer which may be polymeric. So you see, it is always a combination of number of factors, which is it's the third generation of nanocarriers or smart carriers we're talking about. So the typical organic base will include the solid lipid nanoparticles, where you have the liquid which is emulsified and made into small vesicles. And then you have the liposome. Liposome will have this lipid bilayer. Then you have the dendrimers, where you have this symmetric architecture extending uh, in an ordered fashion. They are very good for the drug delivery because they are in a very ordered arrangement of structures. Then you have polymeric nanomaterial. And then apart from that, you have the polymeric and surfactant micellar vesicle system. So let us look into a few of the examples before we go into my way we are talking about, or way we are working on. So how do you design a nanocarrier, which if it is a solid lipid uh, nanoparticle, then you see how it is designed. You have the surfactant, you have the co-surfactant, and you have the lipid matrix, which is supposed to be solubilized, and you have the drug loaded in the core of the lipid matrix. Then you have the liposome. Liposome is again a bilayer, lipid bilayer. The central core may be aqueous. Usually it is aqueous. So if you have any hydrophilic drug, then liposome system is the best method to have it delivered. So you can have the hydrophilic, and a drug can be outside, and the hydrophilic drug can be here, and the lipophilic drug can be outside, because this is the lipid bilayer, and here you have the enclosed aqueous medium. Then this is the dendrimer. If you see, it's a it's a very structured arrangement. You have a core central uh, system, and then you have the terminal groups, which is going to be enclosing the drug molecules very in a systematic manner, and the release is also very systematic. So if you have a dendromitic system, then you have a very good controlled delivery of the drug. Sorry. How do I go up? And then you have this polymeric system, or surfactant vesicles. Yeah, here you have the hydrophilic shell and the hydrophilic segments, and you have the coatings and number of coatings. Whether you use the biocompatible polymers to generate it, you can use surfactant to create micelle. In addition, you have also have this virus systems which can be used to deliver drugs and other lipid moieties. So they are also, their plant viruses are used for delivery as a delivery system. Then you have the carbon based system, which can be fullerene, it can be carbon tube nanotube in the single or multi wall. You can have the uh, graphene oxide, you can have the graphene quantum dots. These sort of things can be talked about, or it can be taken up when we are talking about carbon based delivery system. In fact, this is what I am currently interested in. Now, when I've talked about so many other things, it is high time I talk about certain things and what are the processes I have followed. See, there is no unique way of following preparing a delivery system. I can only discuss what I have followed and what has been published from my group. So the first thing which we had taken was a hydroxyapatite, and we were targeting the colon cancer, and colon cancer you have the folate receptor. So if you want to have this targeted, then you can actually have a folate receptor to be functionalized on the surface. But what we did here, in this publication, what we did is we had prepared the hydroxyapatite, which was a separate work altogether. Then we had this chitosan. Chitosan is a moiety. You have this functional NH2 functionalization on the surface. If you have NH2 functionalization, then you can attach the, this is a say fluorescent moiety which you attach to the surface on the of the carrier, so that we want a thorough output. We want to see how the cell is moving. So this is what FITC is such a moiety which you attach to the surface of the delivery system. Then finally you enclose the drug, which is then you enclose the drug and you have a moiety like this. And how it is going to drug is enclosed here. Over that you have this functionalization, and then you have the moiety which is going to show you the fluorescence. This is the moiety which is attached to the surface through the NH linkage. This will show fluorescence when you are putting it in the fluorimeter, so you can see or track the movement of the particle, or how you can find out how much it has moved. So this is how it looks and how it is going to be released in that site. It is going to be released by the pH. It is, the moiety is going to be detached in an acidic pH, and whenever you are doing that, whenever you are having a pH triggered release, then what you necessarily look into a moiety which is going to detach at acidic pH, because all throughout the body fluid the pH is around 7. When you reach the tumor site, then the pH suddenly changes to less than 6. So if you have less than 6 micro environment, extracellular microenvironment, that means you have reached the tumor site. So if you want to look into it, then you can look into the reference and see how it was done. I'm not going to go into the details of it. I'm going to take up only one or two details. It is going to be time consuming. And then you had this magnetite. Magnetite is something which is quite often used because of its magnetic property. It can be maneuvered through a magnetic field. So your delivery system can be made to reach the site, even if you do not have a folate receptor. If you have a magnetic probe which is going to be carrying it, you have a magnetic field. The navigation can be through the magnetic field externally. But if you have a receptor, then it is an additional benefit you have. So here what you are doing, you see the design which we are making. We are making first we have made the magnetite nanoparticle. Then what do? We have to functionalize that nanoparticle. So we functionalize that nanoparticle with, say, some acidic OH groups or COH group on the surface. Then we use amine attachment. See the amide attachment in this here. That what have we used? We have used APTAMS to attach, and then what you have this moiety, you have the silica coming from the say trimethoxysilane. You are getting the silane part, so you have the silane, and you have the amide moiety, and then you can attach the folate receptor, and through the EDC and NHS coupling, you get the final product, and this is again going to be reaching the target site, because it is again going to be, even in breast cancer you have this folate receptor overexpressed, so you can have this delivered to a breast cancer site. So this is one of the methods which we have you can see. So majorly this again is going to be detached in a acidic environment. So as soon as the moiety reaches the tumor environment, the drug is going to be detached. So we can functionalize the drug, or we can add the drug after we have functionalized. What are we looking into? We are functionalizing the surface with OH groups, and we are functionalizing with the NH group. We are adding a feature which is going to be showing us fluorescence, and we are doing a coupling with the folate receptor. So that we have a folic acid which is on the surface, we have the amides which linkage. The amide linkage is used not only to maybe enclose the drug, but also to attach the fluorescent marker which we are using. Okay, so this is how you structure or design a particular drug delivery system. This is another one where we have zinc oxide. Zinc oxide, we first prepared, each of the preparation takes time. So zinc oxide preparation itself is difficult because you have to make it hollow. We have made it a template method of making a hollow zinc oxide. And then we have wanted a COOH functionalization. So for that, we have used carboxymethylated chitosan. On reaction, it gives you the surface functionalization with the OH group. Then what you do, when you, here we are trying to target the breast cancer, so we can use the folate receptor, and the drug we are using is paclitaxel. Paclitaxel is used in the loading. So what we have to, how we have to load it? See, we are going to have azide, which is going to be introduced with the folate. So azide folate, this reaction, this step has to be separately performed. And then you have the amine, which is functionalization of amine, so that the paclitaxel is attached. So this is how the azide functionalized zinc oxide will look like. Then you also have the loading of the drug in presence of the alkylated derivative of the folic acid. Here you just note, we have not added any say receptor, we have not added any fluorescent marker. We are adding the see the carboxylic acid groups are there, then the amine functionalization there is there, then we have the enclosure of the nanoparticle. of the drug inside the nanoparticle or attached to the nanoparticle, but we do not have a fluorescent marker. And that is the good point about it, because zinc oxide itself is going to show fluorescence. So if you have designed a molecule or a drug delivery unit where you can reduce the number of attachment by choosing the material appropriately, then also it is a very big advantage. So you can see how the particle size is looking. You just look at the zeta potential, you see it is when you have the paclitaxel enclosed in the zinc moiety, zinc oxide moiety, that means you have functionalized and you have entrapped the paclitaxel in the moiety, that is the blue line. You see the stability is quite high, zeta potential is high, that means the stability increases. And you just look at the drug release profile. The drug release profile, when you are studying the drug release, you see when you are having the pH at 7, that means in the body fluid pH, your carrier is not going to offload the payload which is carrying. But when you are seeing the highest possible payload is going to be generated when you are having the acidic pH. This is near the 6 to 5.2 is the range which we can be close to the tumor site. So if you have an acidic pH, then your release profile is enhanced tremendously. So this is going to, which gives you a output which shows that you can use this material with a good release. Now you see this hemolytic assay which is showing with the control. What you have, hemolytic assay is the breaking up of the red blood cells. This is minimum when you have the drug loaded uh, uh, zinc oxide. But when you have the free drug, if you are using the free drug, then the hemolytic assay is comparatively higher. So that means when you are doing a drug delivery system, designing a drug delivery system, what you have reduced is the toxicity effect of the uh, paclitaxel or the therapeutic agent you are using. Now, here you can see another thing which is very important for this particular work is the photoluminescence shown by the zinc. And the photoluminescence is shown by the zinc oxide. If you see at 5.2 pH, and after variation of time, you see how it is increasing. That means when, as soon as the cell wall, the targeting moiety or the drug delivery unit reaches the cell, which is invades the cell and goes into the inside the cell of the tumor cell, then what happens, there is a rupture of the cell boundary, and you have a very high increase in the photoluminescence. So if you have such a readout, then it is obviously going to be very important even for diagnostic, because not only is it getting ruptured and getting inside the cytoplasm of the cell, but it is also endosomal unit of the cell, but it is also giving you a signal that it is entered by having a very high increase, even a 10-fold increase in the photoluminescence property. So this gives you a very big thorough output of the results which you want to observe, because you do not need to do a fluorescence tagging. Your moiety which you have chosen itself is fluorescent, and not only fluorescent, it is actually acting as a switch. As soon as it's entering the cell system, the endoplasm of the cell, then by rupturing the surface, then there is a drastic increase in the intensity of the the photoluminescence. So that means you have indication that your cell has entered the your drug delivery unit has entered the cell. Now that can be monitored. That can be monitored by using the flow psychrometry or the fluorescence microscope. You can make out how they are through the luminescence. You can see how the light, without adding a biomarker, you have this visible light shown here. And then you see the nanocarrier cellular uptake. I'm only talking about in details, and this others I'm not even talking, because I just need to talk about the functionalization. Other parts can be looked up because every system has its own protocol. So when you are talking about the cellular uptake, how much the cell is taken, the nanocarrier is taken up, then you see with increase in time, you have an increase in the cellular uptake, 3 to 6 hours to 12 hours. Okay, so this is one. And then you can also see the proliferation assay, by which you can find out how and this MDH, and these are actually the cancer cell lines which are being used. So if you look at this also, this is the cancer cell line which we have used. This is breast cancer cell line, MCF7 and MDA, MB231. These are all prototypes of the breast cancer cell lines. So you can see how it is affecting the two cell lines. You can make out here with time how it is increasing, and it is affecting the cells. Similarly, you can proliferation studies actually can be done, and you can see how the two cell lines are. You see this indicated. One is the acidic form of the carboxylate functionalized the zinc oxide. This is only the drug, which is paclitaxel, and the last one is the entrapped drug or enclosed drug or attached drug moiety with the zinc oxide nanocarrier. So if you look at this, you can see the red is the how many viable cells are there. You see the viable cells are going to be minimum when you are having the drug delivery system attached to a drug. Similarly, you can see the ACM pictures, and you can make out how this is the morphology of the nucleus can be monitored as the system and the zinc oxide carrier moves into it. So this can be monitored. You are getting a reflection of how much it is entered or where it is going. So this monitoring is done without attachment of a biomarker from outside. So I'm not going to go into the details of it. You can see this is animal studies were carried out to find out. And finally, what we have seen is the hollow nanocarriers. We have this drug carrying the paclitaxel to the side. We have functionalized it with the folate receptor, and it gives a thorough output of readout thorough output, because we do not need to attach a fluorescent moiety. So this is obviously a very good design. And you have, since we have a switch on switch off switch generated, because of the as soon as the drug delivery unit enters the cytoplasm on the cell, Then you can have the increased intensity, so it's behaving like a switch. This can be used also in diagnostic based experiments. And then you can also, we have also seen that if you have just the paclitaxel, and if you have that loaded within a drug delivery unit, like the one we have designed, then you can have the negative side effects can be reduced, because it is actually paclitaxel is highly damaging to the normal cells. So if you can control the damage, then you have achieved much. Similarly, we have introduced zirconium phosphate system, zirconium phosphate core shell system. Here also we have navigated zirconium phosphate. Alpha phosphate is a layered type of structure. It is a cage sort of a structure. So if you if you can have the zirconium phosphate generated, then it will look something like this. There you can attach the nanoparticle which you have generated, and it will look like it is the way it is attached to the surface OH groups of the iron particle, magnetite particle. All magnetite particle will be having some surface functionality, and OH is usually there. So through the OH functionality, it is going to attach, and it may also be intercalated between the layers. You can see it is a layered sort of a thing. It can get intercalated. So it is some sort of arrangement of core shell is generated, because each of the nanoparticle is going to be surrounded by, or they are going to be enclosed in a sheet of the zirconium phosphate. Now you can put the a drug. Here we have taken curcumin as a model drug. Curcumin is not a drug, but we have used it as a model drug because its structural features similar to the drug. So we have used curcumin as a model drug, and we have done that for a number of experiments. So what we have this curcumin attached or intercalated into the structure. So it will be a core shell structure where you have the interactions. It is not may not be covalent interactions, but it can be other type of interactions. Covalent interactions may not be always possible. But even if you are taking this and you see how it is decorated, and this nanoparticle, if you are passing it through a bloodstream and it is reaching the target site, and can we have it as active receptor, active delivery? We have not added a receptor here, or a ligand which is going to be attaching to the receptor which is expressed in the cancer cell. So we have not decorated that with the folate receptor which could be used. So we have just kept studied the drug release studies of curcumin when you have a structure like this generated, a core shell structure of the core being iron oxide and zinc phosphate, zirconium phosphate being the outer core. And this is how the release profile shows. When you have the curcumin and curcumin loaded, you can see the release profile, cell viability profile. Cell viability is going to be maximum when you do not have any drug. And when you compare the curcumin and the curcumin which is enclosed in the zirconium phosphate matrix, or core shell matrix, you see the difference in the biofunctionality of the two, or bioactivity of the two. Then comes, you can look into the how the efficiently drug is loaded, and how efficiently the profile is going to be there. And what is going to be important, that the release profile should be very good in, at least in the acidic pH, and it need not be good in the non-acidic pH, like in the body fluid pH. If it is not releasing in the in the body fluid conditions or pH, then obviously you can at least safely take it to the site where through EPR effect by accumulation outside the epithelial cell, it can act as a therapeutic agent, or it can benefit from the therapy or increase the the efficacy of the drug. I'm not going to go into the detailed profile. It is meaningless until you do it yourself. So obviously you have a magnetic material which is not very highly magnetic, because it is a super paramagnetic particle you have generated. The loop is not opening, so the magnetization is going to be low.
Professor Pathak,
yes,
sorry
yes.
for the interruption. I think we are running out of time. So, maybe.
Okay, I have 2 more slides, I think.
Okay, ma'am. Yeah.
So this is another which I have done with zinc phosphate. Zinc phosphate, again we are attaching here again with the amine functionalization. Then you are doing the EDC and NHS coupling and getting the functionalization, and then you can generate couple it with the drug and do the delivery. This is an isosome system, but I'm not talking about it because I'm talking about inorganic structures, so I'll skip it. These are the inorganic hybrid systems which we have developed so far, and there are a number of them, at least 12 of them, and they have to be unique if they are to be published. So they are all unique in their own way, and these are my collaborators and these are my students. So thanks to their help, and obviously DST and ID Kharagpur for the funding. And before I end, I just want to say that we cannot have a unique system generated. I cannot say that what I have done is the best, or whatever is available is the best. There is still no universal platform which is going to be suitable for all kinds of drugs and for all kinds of types of cancer. So it is not one fits all sort of a condition. So whatever we have done, there can be improvement, there can be better conditions which you can generate, but always every drug and every uh, type of cancer or tumor which we are looking into is going to have its unique conditions, and accordingly you have to decorate the surface. Thank you so much.
Thank you, Professor Pathak, for this wonderful presentation and sharing the interesting research you are doing with these drug delivery systems. Thank you very much. I think we are running out of time. So, because at 2 o'clock we will be having these oral presentations coming. So maybe we can take one quick question if anybody has
Everybody
something to
is
ask.
hungry. Everybody is hungry. They have left already. They can send
Yeah,
me
I see
questions,
people.
because actually I do not work in drug delivery anymore. So
Okay.
I can always take up the question. But for the last 4 years, I've shifted my research field.
Okay. Okay. So I request the participants, if you have any questions, feel free to contact Professor Pathak. She'll be happy to answer your queries. With this, I think we all thank Professor Amita Pathak for her time and for her lecture. Thank you. Thank you very much, Professor Pathak.
Thank you so much.
It was nice to have you with us. And
Yeah.
then with this we will close the morning session. So we can all meet again at 2 o'clock with the oral presentations. Just to remind you, we are having 3 parallel oral presentations running in 3 different streams today. We have organic and biomolecular chemistry stream. which will be chaired by Professor Jaikumar Kandaswamy from IIT BHU. And then we have nanomaterial stream, which will be chaired by Professor M. Led by Professor Isa Trivandrum. So with this, we will close the morning session. I'll see you all at 2 o'clock. Thank you. Thank you very much, one and all.
Yeah, oh yes, you got it. But yeah, yeah. So I don't know. I didn't reply. There is an offer letter. There was date mark and
What are you doing?